Hello, my name is Jenny Ricker, and I will be conducting a DNA extraction experiment. DNA stands for deoxyribonucleic acid. It is a molecule that is composed of nucleotides that always include the following. They always include sugar, they always include phosphate, and a nitrous base. The importance of DNA is pretty vast. I can just name a few. They are responsible for delivering genetic information to every single cell in our body. They aid in heredity and also the creation of species, embryo development. The list goes on and on. So with the experiment, I have taken the liberty to have pre-blended the peas with the 1 cup of distilled water and the salt provided. We will now be taking this mixture and draining it through a cheesecloth. Little technical difficulty there. Okay, all right, great. So we have now, what is underneath the cheesecloth is DNA. So I'm going to carefully remove the cheesecloth, okay? Discard it elsewhere. We are now going to add the provided detergent and then use a stirring rod to stir it for about 10 minutes. So it's been 10 minutes now. So I've taken out the pea detergent water solution, and I have poured 1/3 of the solution into this test tube, okay? From here, the next instruction is going to be to add about half of the meat tenderizer powder directly to the tube. So, take care of that. Okay, I'm going to now use the stir to stir in the meat tenderizer. Okay, so this has been sitting in my freezer overnight. This is the 91% proof alcohol. I'm supposed to fill this to halfway of the actual pea solution at a 45-degree angle. Okay. It's brand new. Okay, let's see here. Maybe I could do. Nope. Okay. Dissolve. Okay, so gently pour this in at 45 degrees until it reaches the level of being halfway. What do we say? It's a little bit more, but it's pretty close. Pretty close. Okay, so from here, so now we are ready to make observations of the DNA, which is the white that has formed, and it's definitely gotten more prominent since the addition of alcohol. We're going to compare the 2 pea samples to see the color differentiation. So it's actually remarkably the same. It's just that obviously in this one without the alcohol, the DNA strings are less. So this concludes the experiment. I would like to address a couple technical issues or reasons why we included certain items. We included salt because it breaks down the cellular membrane, making DNA much easier to be released. I believe the next question was, why did we add papillon powder? Now, that's an enzyme from papaya, which also helps break down the cellular membrane for DNA release. Finally, why did we use 91% alcohol? DNA does not dissolve in alcohol, so alcohol aids in making DNA's presence more visible. I hope you enjoyed this experiment, and I have a great day. Bye. I'm back for an encore. I just wanted to touch upon a couple things that I think I missed. First of all, sorry that the test tube was not shown as prevalently as the glass. So I want to just talk about, again, describing what we're seeing here in terms of how thick the DNA is and stringy it's getting, and how the example without alcohol has minimal DNA. The colors go from dark to light, and that's true for this as well. Okay, then also in regards to materials that we used, I forgot to talk about detergent. Detergent is what we use to also help dissolve the membrane. So I think I've covered all of the questions. I found this really fun to do, and yeah, have a great day. Bye.